Hello, everyone. So my name is Yue Zhao, and I'm a research assistant professor. So my research interest is mainly focused on genomics and transcriptomics research, and especially in neuron degenerative disease and cancer. So today I will present one tool developed several years ago, and its name is called the CEDA. It's an R package, and it's integrating gene expression data with CRISPR screen data. And the goal is to identify essential genes with higher expression level. So first, I will briefly introduce the CRISPR-Cas9 system. So as you may know, CRISPR-Cas9 system is initially a microbial adaptive immune system, and it's later on developed as a powerful genome editing tool and won the Nobel Prize in 2020. So this system composed of two parts. One is the guide RNA. Initially, it has two parts, the tracer and the CRISPR RNA, but later on, people just combine them together to become one single guided RNA. So the 5' end of this guide RNA has 20 nucleotide sequence that is reverse complementary to the targeted site on the genome. And this guide RNA direct the second part, this is the Cas9 endonuclease, to cut the target genomic region. That's a double-stranded card. And then the subsequent DNA repair via the non-homologous and joining often introduces small insertion and deletions that can induce a coding frame shift or a premature stop codon. And those artificial genomic variations eventually lead to non-functional protein or degraded messenger RNA, and therefore a functional knockout. So other genomic perturbation, in addition to this functional knockout, can be achieved by fusing the deactivated Cas9 endonuclease with different functional domain. For example, to inhibit transcriptional activity, like in CRISPR inference screens, CRISPR-I, or to activate the transcriptional activity, like in CRISPR activation screens, CRISPR-A, or it can edit specific nucleoside, like in the base editor screen. So in the case of the growth-based negative screen, this we can say is the most common or most basic CRISPR-Cas9 genetic screen. So we usually start with millions of cells, and we first introduce the lentivirus with the guide RNA into cell, and each cell, each guide RNA will enter to one cell. That's what we achieved by controlling the MOI ratio. And then this is our baseline E0 sample. And we collected those DNA and send them to sequencing. And after a few weeks, usually after 3 or 4 generations, then we collect the endpoint sample and also extract the DNA sent to sequencing. Then we compare the guide RNA recounts in these two samples. So here, each guide RNA, the recount present one cell. And then if there is a reduced number of the guide RNA, like in the dark blue and the light blue cases here. So that means those cells reduced, or they will be died after knock out the gene that this guide RNA targeted to after the cell proliferation. That means in the normal case, the targeted gene is a cell growth inhibitor, or it's an essential gene for the cell to grow, because knocking out this gene, the cell will die. And in another case, like in this yellow bars here, the number increased after cell proliferation. That means in the normal case, this gene will be like a cell growth inhibitor, or in the tumor case, it will be a tumor suppressor, because knocking out it, the cell will grow happily and will grow more. And it depends on the type of the library construction. we can have single knockout screen, like I show here. Each guide RNA is encoded on a single construct. Or we can have a double knockout screen. Here, two single guide RNA are encoded on a single construct. And usually these two guide RNA are targeting two different genes. So ideally, each cell will have two functional knockout after introduce those lentivirus. And based on different phenotypes, we can have dropout screen. For example, a growth-based screen here can identify essential genes that influence the relevant cell viability. Or we can have a non-dropout screen. For example, here is a differentiation screen, and we use some cell sorting tools to sort cell type based on the marker gene. And we can compare the guide RNAs in type 1, type A, and type B, two different cell types. So the computational analysis for large scale, the CRISPR screen usually start with the sequencing read data. We can have a tabular data with each row corresponding to each guide RNA and each column corresponding to each sample. And after getting this tabular data, we can do some aggregation analysis to aggregate the guide RNA level information to gene level. Remember, we usually have 4 to 6 guide RNA targeting one gene. So we need this aggregation step. And then we can plot this gene effect analysis with gene rank in the x-axis and the gene effect size, or the aggregated log fold change of each gene on the y-axis. So the negative controls usually have the no effect size, and we can have depleted genes with negative log fold change or enriched genes with positive log fold change. And then if we have double knockout screen, we can study the genetic interaction analysis. And the downstream analysis can involve like a pathway analysis or network analysis. So here I just listed briefly in the past 10 years, there are over 20 different bioinformatics tools developed for large scale CRISPR screens. But in this talk, I will just briefly introduce two of them. That's the two most popular ones. The first one is called BAGEL. It's a short for Bayes analysis of gene essentiality. So we have gold standard reference sets for the essential gene screens. Actually, we have more than one such set. So the first set is developed from Morpheus lab, and it's derived based on the RNAi screen of the 48 different cancer cell lines. It contains over 600 core essential genes and over 900 non-essential genes. The second essential gene list is developed by the Magic Flu paper in 2017. It contains more than 600 essential genes and 350 non-essential genes. Those are identified based on CRISPR screens performed in 66 cancer cell lines. And the third list is the ribosomal protein genes. Because we know the ribosomal protein genes play a very important role in cell growth or cell proliferation, and their knockout is likely to be lethal. So they are considered as the third list of the core essential gene. And as you can see here, the overlap between the essential genes are not very good. There's only 31 of them in common. However, in the other side, the non-essential gene lists are quite consistent. And let's go back to BAGEL. What the BAGEL does is it uses the prior distribution of the non-essential genes. That's the non-effect here. And also the positive effects, that's from the essential gene list. So BAGEL utilized the prior information of these two core essential gene list and non-essential gene list to calculate the Bayes factor for each gene. And then they rank the genes according to the Bayes factor. So BAGEL essentially is a gene essentiality classifier. and it performed really, really good in classifying the essential genes or non-essential genes. However, it has limited application in a broader range of screen, like a CRISPR-I screen or CRISPR-A screen. It doesn't perform well. And the magic RNA is the first method developed dedicated specifically for CRISPR screen. And it has two modes. I will first introduce the RNA mode or the robust ranking algorithm mode. So in the magic RNA mode, the mean variance relationship of the guide RNA reads are modeled by the negative binomial distribution. This is very commonly used for the sequencing reads. And then they use the modified robust ranking algorithm to combine the guide RNA level p-value to get the gene level p-value or the gene rank. Finally, they calculated the FDR for each gene from the permutation p-values using the Benjamin-Haukeberger procedure. And the second mode of the magic is called the magic MLE. So in the previous mode, the method can be only applied to comparing two conditions, and it didn't consider the guide RNA knockout efficiency. In the magic MLE mode, it improved based on the alpha RNA, so it can detect the target gene across multiple conditions, such as in different cancer cell lines, or different drug treatments, or different time points. And it also addressed the variability in the guide RNA knockout efficiency. So the knockout efficiency and the gene effect size can be estimated at the same time by the EM algorithm. So I'm showing here, it's just one application of this magic MLE method on a kilobyte CRISPR-Cas9 screen I did in my research. So we started with 3,000. Let me first say the purpose of the goal of the research is because the prostate cancer will eventually develop a drug resistant to the enzyme treatment. So we want to identify if there is any gene that can be potentially be a drug target to resensitize those prostate cancer non-responders. So we start with over 3,000 guide RNA targeting the human kinase genes, and then we introduce the lentivirus into those cells. And the cell line is 22 RB1 cell line, which is known to be intrinsically resistant to the drug enzyme treatment. And after the antibody selection, we only keep the cells with guide RNA. And after 2 weeks, we collect the sample. The DMSO is the control sample, and the enzyme drug treatment is the treatment sample. So, and then we apply the magic MLE and calculate the beta value for each gene. Here, you can consider the beta value as the log fold change. So ideally, we want to find some gene that in the right bottom corner here, because we want to have those cells that died sooner than the control in the drug treatment cells compared to the control cells. That means those cells with that gene knockout become sensitive to the drug treatment instead of resistant to the drug treatment. So we want this beta score difference, the treatment beta minus the control beta, to be most negative. Those are the genes that we are mostly interested in. So it will be in this order. And indeed, we found 2 interesting genes that is known to be very interesting in the prostate cancer. The first one is mTOR, is a known drug target in the prostate cancer. And the PLK1, its expression is known to be linked to the prostate cancer, a tumor grade. And the third gene is what we discovered. It's called the CINK1A1. And it's show a very negative score and it has never been studied in the literature. So we went on to do further validation test in the cell line, 22RA1 cell line, and we developed a gene signature that is 188 gene signature that is overlap this CT1 alpha overexpressed genes and the CT1 alpha knockout upexpressed gene in the CT1 alpha knockout cells compared to the control. So CT1 alpha is the protein that this is CSNK1A1 coded. And then we further validated this CT1 alpha negative signature indeed is negatively enriched in the drug non-responders. That means this CT1 alpha level is highly expressed in the non-responders, and inhibiting this CT1 alpha protein or the CSNK1A1 gene expression will resensitize those it will potentially resensitize or overcome this enzyme resistance for the prostate cancer. So here I'm just showing a very example how we start with a CRISPR drug screen, and we can follow the exploratory results and to further validate or test some hypotheses or have some novel discovery. And then in another study, we ran this magic MLE mode method on a breast cancer cell line. It's MDA-MB231 breast cancer cell line. And we found a very interesting observation that the identified genes were enriched in highly expressed gene. Or we can see here the x-axis is the gene expression, y-axis is number of gene. The orange color is the gene, number of genes selected by the magic method. And we can see with gene expression level increase, we have more number of genes selected. So this tells us that the selection signal is very strong for highly expressed gene, while it's very noisy for lowly expressed gene. And this motivates us to considering combined gene expression data may benefit our CRISPR screen results. And we also noticed that there are other bioinformatics challenges, even though we have some computational advances, but there remain some challenges. So first is existing tools usually have loose positive control and output a long list of candidate genes. And the second is remain challenge to identify genes with moderate effect size in guide RNA fold change. And these genes can be important in target discovery. And the third one is existing computational approach tend to have problematic prediction of genes with extremely low or no expression. So these genes are hard to be white lab validated. So therefore, we developed CEDA, is short for CRISPR screen with expression data analysis workflow. And thanks for my collaborator, Dr. Lianbo Yu. He contributed a lot for this method development. And the CEDA workflow contains 5 parts. The step A is to normalize guide RNA counts and calculate the log fold change by lemma. Step B is to split the log fold change into bins by gene expression. So here we started introducing gene expression data. Step C is to fit a hierarchical model with the normal mixture. And then we compute the posterior log fold change by an empirical Bayes method and obtain the p-value for each guide RNA. And the last step is to calculate the gene level p-value by the alpha robust ranking algorithm and calculate the gene level log fold change by averaging the guide RNA level log fold change for the same gene. So I will briefly go through some mathematic equations. Bear with me. It will be pretty fast. So we model the guide RNA recount to actually the log2 transform the guide RNA recount to follow a normal distribution with mean mu and the variance sigma square. And the mean is the sum of the baseline beta I0 and the beta I1 is the mean difference between the two conditions. Since we log2 transform this recount, so we can consider beta I1 here, you can consider it as a guide RNA log fold change between the two conditions. And our testing hypothesis is whether this log2 fold change is 0 or not between the two conditions. And then we started to introduce the expression data. Let EG to denote the expression level of the gene. And then the range of the EG is divided into 5 intervals. And we introduce a Latin variable and assume it follow a multinomial distribution. So the log fold change will be 0, will follow a normal distribution with mean 0. For this is representing the equal representation case, like there's no change. This is non-essential gene. And then the beta I1 will follow a normal distribution with a positive mean alpha 1. That is for the enrichment case. And then the beta I1 or the log fold change will follow a normal distribution with a negative mean value. That is representing the depletion case. So then we use the empirical Bayes estimation method to estimate the hyperparameter. So first, the no change for the no effect for the non-distribution. The variance for the non-distribution is estimated by the sample variance, and then the starting value of those model hyperparameter is also estimated by the sample means or the sample variance. So here is the EM algorithm, the E step is quite standard, so I will just skip it. And then that is the M step. So finally, the number of mixture components are estimated by the information criteria. So for example, here in the first 2 bins, bin 1 and bin 2, there are 3 components. And in bin 3, 4, and 5, there are only 2 components. And then because the assumption of the conjugate prior on the log fold change, the posterior log fold change is also normally distributed. And the p-value for this testing hypothesis can be obtained by evaluating the posterior mean of the log fold change under the non-distribution. So, okay, after going through the detailed method, so we started to evaluate our method. To demonstrate the benefits of using the gene expression data for the log fold change shrinkage, we evaluate the genes selected by our method using the gene expression data. That's the red dots here, compared to not using the gene expression data. That is the orange dots here. So, the blue one is the common one. Of course, both of them are common. They're just a few red dots and even fewer orange dots. So here I highlighted the 5 genes that with this black circle. So let's start with the BRCA1. BRCA1 is known to has a harmful effect on MDA, MD 231 cells. And then this LSM12 and the CFL1, they are known essential genes in the actually in the BAGO2 essential gene list. And the other 2 genes, MRPS6 and MRPL39, they are also known essential genes in the magic flu essential gene list. So in the other words, these 5 genes would be missed if we were not integrating the gene expression data with the screen data in our model. And indeed, the percentage of essential gene increased when we integrating the gene expression data. And also use the gene expression data, select the gene. They are having higher expression values. So next, we look at the control of the false positives. Again, we use the core essential gene and the non-essential gene I mentioned earlier, and to test, compare our method, CEDA, with magic in RA mode and magic in MLE mode. And the G-screen is another popular method in the CRISPR screen data analysis. So here, ideally, okay, I'm plotting the core essential gene list, non-essential gene list here. And then the y-axis is the FDR of the selected of all the genes. So ideally, we want all the genes fall below the 0.05, this selection cutoff for all the core essential genes. And we want, there's no dots here for the non-essential genes because we don't want to wrongly select any of those non-essential genes. And as you can see, out of all the 4 different methods, CEDA is the only one that correctly didn't select any non-essential gene. So that demonstrates CEDA has the best false positive control. And we also look at the effect size, the gene level log-fold change of these 4 different methods. And you can see the CEDA has the best separation between those genes with different FDR range, especially for the selected one. That's the purple and the green is the FDR less than 0.05, and the other 3 colors representing the FDR. Sorry, the other way. The purple and the green is the FDR greater than 0.05, and the other 3 representing the selected FDR less than 0.05. And they have pretty good separation in the effect size. And we also look at the gene expression level. And we also similarly stratify all the genes by FDR range, and we plot for each stratified gene groups. And CEDA is the only one that didn't select any genes with low expression, and all the others have some noises or the low expression of the gene selected. So we further compare the top 500 genes ranked by each method. So there are over 200 genes were commonly selected by all the 5 methods. And those are the yellow ones here. And you can see they tend to have large log-fold change. And this is easy to understand because genes with larger log-fold change is easy to be identified as like selected genes. But I highlighted the genes selected by one method only. And those red dots are the 55 genes selected only by CEDA. And those genes, as you can see, they tend to have moderate or small log-fold change. So those are hard to select, but the CEDA can pick up those signals. And to further understand the difference produced by this method, we look at into 2 specific case, 2 essential genes that are only selected by CEDA but missed by most of the other methods. And I plot the guide RNA change at T0 and T1. And we can see they are not 100% consistent because, for example, this guide RNA show a different trend compared to the other 3 guide RNA. But however, the majority of the guide RNA still show a pretty consistent depletion or strong depletion at the end time point. So we can see we are quite confident that we are selecting the correct gene. So let's go back to the question. So why integrating the gene expression data can help with the CRISPR screen selection results? The motivation of our study is the observation that the selected genes in CRISPR screen, the number of selected genes in CRISPR screen tend to increase with increasing gene expression. This kind of give us a clue, or this could be potentially explained by the positive association between the gene expression and the chromatin accessibility. So genes with high expression usually locates in the regions of low nucleosome occupancy and high chromatin accessibility. And interestingly, highly active guide RNA for Cas9 and the deactivated Cas9 nuclease also found almost exclusively in those regions. And the Cas9 cleavage or Cas9 activity is also strongly inhibited when the DNA target is within the nucleosome. And this gives us some hypothesis or some potential explanation why integrating gene expression could help. So that's maybe because the guide RNA efficiency and the Cas9 activity information can be indirectly provided by the gene expression data. So taking advantage of the prior information from gene expression data could benefit the selection results. So in summary, CEDA is a tool that integrates gene expression data for CRISPR screen and to identify the target gene with higher expression. And what the CEDA does is to stratify the guide RNA log fold change based on the level of gene expression data. And it can properly control the false positive rate and identify essential genes with higher expression and the moderate effect size. And this R package CEDA is available to download at the CRAN repository. So finally, I want to acknowledge the co-authors and the collaborators of this paper, Lianbo Yu and Dr. Lang Li, Snow, Calvin, Haoran, and also Yunhao, Qinfei, and Li Jun. Okay, so now I'm open to any questions. Yes.
Fantastic. Does the gene expression data that your method used have to come from the screen itself, or can you use like the cancer cell line encyclopedia gene data or phenotype?
Yeah, we just use the RNA sequencing data from the same cell line, but we also tried to use the cell line data, like the available cell line data is also okay. Actually, it's a very interesting question because we were thinking, should we use the time zero RNA-seq data, gene expression data, or T1, the end time point data? We are thinking maybe we should do some comparison between these two. But now currently we just use the baseline time zero data. Yes.
Great talk. I have two questions. First, when we talk about the essential gene, knockout gene that the cell will die, we call it essential gene. But you know, there are two or several studies, the essential gene, the overlap is not good. So I think about when we thought about the essential gene, it should be robust to train the different studies. So what's the reason to it's not robust across the different studies?
Yeah, that's also a very good question. I was also very feel interesting. The non-essential gene, the overlap very good, right? But the essential gene, the overlap is just 31 out of hundreds of the essential genes. So I think one is the technology. So one list is generated based on the RNA inference screen, from the MOFAT, like the Babel2 version essential gene list. The second essential gene list generated from the Magic Flu is from the CRISPR knockout screen. So I think the fundamental technology difference will introduce a lot of difference in this. The second one is also the cell line. They are based on different cell lines. I didn't look at whether the 48 cell line and the 66 cell line, how much they overlap with each other. I didn't look at that, but I think that's also cell line dependent.
Okay. Second question is, you know, the sgRNA, the CRISPR, there are some positive, possible to off target. So how could we avoid this kind of target effects? They are also important.
They are important. They are very important actually. But that's something I think we need some technology improvement. And for the CEDA, we didn't consider this off-target effect. And that's a big limitation. Yeah, I have to admit. And I like a magic MLE that they can input the guide RNA efficiency and they can incorporate it into their method. Yes.
So there's a lot more going on in cancer cells from a genomic perspective than just the expression differences. There's copy number changes and mutations. Could you integrate that information, which is easily gettable, to also improve the correction you're doing here?
Yes. That's actually an ongoing project we are doing now. We are trying to incorporate the SNP information or copy number information into this. It's a very good point.